We often think of a heart attack as this, single devastating event. And, well, of course it is.
Absolutely.
But what really dictates long-term heart health is often what happens afterwards in the days, the weeks that follow.
Right.
Today, we're doing a deep dive into some fascinating new research that's really shifting our understanding of that ongoing damage after a heart attack.
Yeah, it's like finding a hidden mechanism we didn't know about.
Exactly, like unlocking a crucial puzzle piece.
And it's so important because ischemic heart disease, which, often starts with that heart attack, myocardial infarction, or MI.
Blockage.
Exactly, when blood flow gets blocked, it's still the leading cause of death globally. So understanding what happens after that initial hit, the damage processes, is just vital.
Absolutely. And, well, for a long time, the main idea has been that the continued damage is mostly the immune system.
Yeah.
Right, rushing in to clear out the dead heart tissue.
Mm-hmm. That's this sort of classic view, the body's cleanup crew, as you said.
Yeah, like first responders dealing with the immediate aftermath.
It makes sense, intuitively. You have cell death, debris, and the natural response is for immune cells, neutrophils, macrophages to come in and handle it.
But this new work from UC San Diego, Dr. Kevin King's team.
Yes, exactly, at the Sulpizio Cardiovascular Center. Their research, it was just published in Nature, suggests the initial trigger for inflammation might be, well, something quite different, something earlier.
Okay, let's unpack that. So forget just the immune cells swarming the wreckage for a moment. What did Dr. King's team actually find kicks things off?
So they identified this really significant inflammatory response, specifically the activation of something called type I interferon signaling.
Type I interferon. Okay.
Now, what's really fascinating here is where this was happening most strongly. It wasn't right in the zone of dead tissue, the infarct core.
No. Where then?
It was most active in the borderzone. That's the area of surviving heart cells right next to the damage.
Ah, the cells that made it through the initial attack but are still right there on the edge.
Precisely. These are cells that survived, but they've experienced this massive shock, this change in their environment. They're stressed.
The border zone, that sounds like a really critical area, kind of this in-between space. But I imagine it must be incredibly difficult to study because it's such a mix.
It is. Traditionally, it has been. These surviving heart muscle cells, cardiomyocytes, they're under huge mechanical stress. They're trying to adapt, maybe even repair things.
And the researchers use some pretty advanced tools to look at this.
Oh, yeah. Incredibly sophisticated stuff. Single-cell RNA sequencing and spatial transcriptomics.
Okay, what does that let them do?
Think of it like getting a readout of which genes are switched on or off in every single individual cell. And not just that, but where those cells are located within the heart tissue itself.
Wow, so incredibly detailed, like a molecular map.
Exactly. A much, much finer level of detail than we've ever had. It allowed them to actually identify these specific border zone cells based on their unique gene activity patterns, getting around that problem of physically isolating them.
So, okay, inside these stressed but still living border zone heart cells, what was the surprising trigger they found?
This is the really remarkable part. These mechanically stressed cardiomyocytes, they were actually experiencing something called nuclear envelope rupture.
Nuclear envelope rupture, so the lining of the nucleus where the DNA is kept?
Exactly. Imagine the cell's command center, the nucleus holding the DNA, actually getting physically damaged, like torn, because of the mechanical strain it's under.
And that damage lets the DNA leak out.
Yes. DNA spills out from the nucleus into the main body of the cell, the cytoplasm.
And the cell detects this escaped DNA, like an internal alarm.
Precisely. The cell has these built-in sensors designed to recognize DNA where it shouldn't be outside the nucleus. It thinks it might be, say, viral DNA.
Ah, okay.
So this misplaced DNA triggers the activation of that type I interferon signaling pathway we mentioned. That's a key part of the inflammatory response.
So wait, the surviving heart cells themselves are kicking off inflammation before the big immune wave even gets there.
It seems so. They appear to be a major and perhaps the very first source of this specific type of inflammation right after a heart attack.
Wow. That really changes things, doesn't it? We've been so focused on the immune cells responding to the dead tissue.
It's a significant shift.
And now it looks like the surviving stress cells are actually initiating a big part of the inflammatory problem themselves. It's like the house is damaged and the people still inside are accidentally setting off the fire alarms.
That's a great analogy. It really is. And this has really important implications, obviously.
How so?
Well, the researchers used genetically modified mice, too. They had different groups where they could switch off this IFN signaling in different cell types, heart muscle cells, immune cells, etc.
Yeah, what'd they find?
They found it was the heart muscle cells, the cardiomyocytes, that were the primary drivers of this specific borderzone inflammation. And crucially, this inflammation led to a weakening of the heart wall,
Weakening how?
making it more likely to dilate, stretch out, get thinner, even rupture in severe cases. Interestingly, the mice that couldn't mount this IFN response in their heart muscle cells, they actually had better survival rates after a heart attack.
Okay, so preventing that specific inflammation pathway in those specific cells seems protective.
It points strongly in that direction, yes.
So what does this all mean for, how we might actually treat heart attacks to prevent heart failure down the line?
Well, connecting the dots, it really points towards entirely new potential therapeutic targets, things we haven't focused on before.
Like what?
Instead of just trying to broadly dampen the entire immune response, which honestly hasn't worked very well with traditional anti-inflammatory drugs in this context.
Right. Those trials haven't panned out so well.
Exactly. So maybe we could focus instead on reducing the mechanical stress specifically on those borderzone cells.
Tackle the initial trigger.
Precisely. The idea would be to prevent that nuclear envelope rupture, stop the DNA leakage from happening in the first place.
That makes a lot of sense. If the spark is the stress causing the DNA leak in those cells, targeting that stress seems much more direct.
It's a much more upstream target. Additionally, we could explore ways to block those DNA-sensing pathways within the cardiomyocytes.
So even if DNA leaks, the cell doesn't sound the alarm.
Potentially. Or we could look at inhibiting the type I interferon signaling itself, but again, specifically within these heart muscle cells.
Much more targeted approaches compared to just general anti-inflammatories.
Absolutely. And the fact that those broad drugs haven't worked well might, in hindsight, suggest we were targeting the wrong type of inflammation, or maybe the wrong source, the wrong cells, or perhaps just too late in the process.
And the fact they could pinpoint this using those advanced techniques, the single-cell stuff, it really shows how vital those tools are becoming.
Oh, completely. Being able to study these very specific microenvironments within the heart is crucial.
It's like these new tools are giving us a level of insight, a granularity we just couldn't get before.
Absolutely. The border zone was always this kind of black box, a bit mysterious, but these single cell and spatial technologies are letting us finally map out its complex cellular and molecular landscape in really unprecedented detail.
Which you need if you want to develop these kinds of targeted therapies.
Exactly. You need to know precisely what's happening, where it's happening, and in which cells it's happening.
So to kind of boil it all down for everyone listening, the huge takeaway here is that after a heart attack, it's not just the dead tissue and the immune cleanup crew causing problems.
Right.
It's also the surviving heart cells right there in the border zone getting mechanically stressed, their nuclei literally breaking open a bit, leaking DNA.
Triggering their own internal alarm system.
And kicking off a significant inflammatory response themselves. And this self-inflicted inflammation then contributes to more damage, potentially leading to heart failure. It really feels like finding that missing piece,
That's the core of it, yeah. This unexpected discovery really shifts our focus. It's not just about the dead zone, it's about these stressed, surviving cells right next door. They're emerging as a primary, maybe even initiating source of inflammation right after the MI.
And this new understanding, it just opens up completely new avenues for potential treatments, moving away from those broader, less successful anti-inflammatory drugs,
which haven't really delivered,
towards much more specific strategies. Like, could we reduce the mechanical stress on those borderzone cells? Could we block the DNA sensing inside them? Or inhibit that specific IFN signal just in those vulnerable cells?
Exactly. Those are the kinds of questions researchers will be tackling now. Of course, there's still a lot we don't know, many questions to answer. Future research is definitely needed to fully understand these pathways, and importantly, how to safely manipulate them for therapy.
Sure, it's early days for treatments, but the direction is promising.
Very promising. It offers a genuinely new direction for trying to prevent that devastating progression to heart failure after someone has a heart attack.
It's truly fascinating, almost mind-boggling, to think that our own cells, the survivors, in their struggle to adapt after something so traumatic, might inadvertently be causing more damage like this.
It highlights the complexity of biological responses.
It really does. And it makes you wonder, doesn't it? Given this new insight into how our own internal cellular processes can have these surprising triggers, well, what other biological mechanisms might have similar unexpected initial events that we're only just beginning to uncover?